Let's begin. Arabidopsis thaliana is a small annual rosette herb native in areas of western Eurasia. It is commonly found in disturbed, poor, stony, or shallow soils, specifically located in nutrient poor, often sandy, meadow, and forest habitats. It is widely used as a model organism in plant biology. Though it may not have much of an agronomic significance, it can provide important advantages for research in genetics and molecular biology. The said plant was discovered by Johannes Stahl in the 16th century and was initially called Pilocelia silicosa. It has gone through many name changes and is now widely recognized as Arabidopsis thaliana, or by its common name tail cress or mouse ear cress. Let me share to you a few fun facts I've acquired. First, it completes its life cycle within 8 to 12 weeks from germination to harvesting. Next, that it has the capability to grow in restricted spaces and is very easy to maintain in an indoor growth chamber. Did you know that Arabidopsis thaliana is a self-pollinated plant? And cross-pollination can also be done easily inside the laboratory? It too produces many seeds. Each silique or seed capsule contains about 30 to 60 seeds, and each plant has around 50 to 60 siliques, so one plant of this species can produce 1,000 plus more seeds.
Thanks, Aya. We will now discuss the taxonomic classification of our model organism, the species Arabidopsis thaliana. It belongs to the domain Eukarya under the kingdom Plantae. Its subkingdom is Viridiplantae, which is characterized by green plants in the infrakingdom Streptophyta, which consists of land plants. The organism belongs to the division Tracheophyta, which homes vascular plants and those of the tracheophytes. Its subdivision is the Spermatophytina. Spermatophytes and seed plants belong in this subdivision. The species is under the class Magnoliopsida and belongs to the order Brassicales. It is in the same taxonomic family as mustards, mutardes, and crucifers, which is the Brassicaceae family. Lastly, its genus is Arabidopsis, and the species name is Arabidopsis thaliana. Next, we will have Troy.
Thank you, Jasper. We will now be moving to the genetic study summary. During the late 1990s, Arabidopsis Genome Initiative began as an effort to decode the genome sequence of Arabidopsis thaliana. This paved the way to significant scientific achievements, and one of its first achievements is in the year 2000s, wherein Arabidopsis thaliana became the first flowering plant to be completely sequenced. Another notable finding is that the genome also contains a similar number of gene functional classifications as other sequenced eukaryotic genomes such as Drosophila melanogaster and Caenorhabditis elegans. The image on the screen is taken from the study of Provart et al., summarizing the different firsts of the 50 years of Arabidopsis research. Some notable highlights include the use in cloning of tDNA and the isolation of plant immune receptor gene. In the year 2004, disease-resistant genes were discovered, which includes the RPS2 and RPP5 genes, which give resistance to Pseudomonas syringae and Peronospora parasitica, respectively. In the following slide, we have other significant findings regarding Arabidopsis thaliana. This includes the study of Mergner et al. In 2020, which quantified the total number of detected Arabidopsis proteins to be more than 18,000. Progress has also been made in the development of different software to further analyze the 1,001 strains of Arabidopsis thaliana. This includes the software of Viva and Arafeno tools. And lastly, new discoveries involving plant immunity were made, such as the full characterization of the Arabidopsis NLR row.
Thanks, Troy. On this slide, we present an experimental research paper that utilized Arabidopsis thaliana. The study of Brazales, Ceballos et al. Tested the abilities of the horseradish Moringa oleifera, specifically the Moringa leaf extract, as a biostimulant for agricultural purposes. The researchers found that MLE has improved the physiological parameter of both seedlings and adult Arabidopsis thaliana, where an increase of analyzed metabolites in the seedlings and significant growth in the total leaf surface area from the adults are observed when treated with the extract. Tolerance to salinity was also detected from the seeds having treated with MLE due to the presence of polyphenolic compounds. Lastly, Arabidopsis thaliana was tested to resist fungal infection of botrytis cinerea when treated with MLE due to its reported antimicrobial properties. Due to the widely accessible data of the model organism Arabidopsis thaliana, studying capabilities of MLE in a molecular level is characterized. Now moving on to our research study proposal, we have Francisco Isidro.
Thank you, Tere, for that wonderful insight. Now we proceed with the research study proposal. So for our proposal, we were able to formulate measuring Desmontes-induced Arabidopsis thaliana towards its inhibiting ability against leukemia in the bone marrow. So what is the basis of our proposal? In a research conducted by Bomer et al. In 2020, Arabidopsis thaliana was induced with Desmontes. The experiment formed a synergetic effect. It was measured to be effective in inhibiting the growth of breast cancer cells while staying neutral or causing no damage to non-tumorigenic cells. So for the proposal background, similarly, leukemia, a cancer that targets the blood-forming tissue, is a type of disease that is widely distributed on non-tumorigenic cells. These non-tumorigenic cells are mostly found in the bone marrow in a form of stem cells. Now, for the research study proposal, the synergetic effect of Arabidopsis thaliana together with Desmontes can be integrated in the cancer treatment against leukemia. The proposal is set to measure if it can inhibit the growth of cancer cells in the bone marrow while causing no damage to the non-tumorigenic cells or stem cells. So for the short schematic diagram, we first proceed with the experimental identification of effects, and the methods to employ here are through plant leaf disc bioassay and Western blotting. We can see here a short illustration above me, a leaf disc sampling. In the research, we were able to retrieve the basis of proposal rather. So for the medium to employ here, so mediums for the methods, we have the co-incubation via incubator. This is for the leaf sampling, and nuclear spin RNA protein kit and protein kit quantification assay for Western blotting. Now, once we are done with this, of course, we proceed with the recording of data and analysis and interpretation. So that's all for the research study proposal. And now here are our list for our references. Okay, and that concludes our report. Thank you.